Hello everyone, and welcome to a presentation on the assessment of cannabinoid levels in successfully cloned generations of industrial hemp. This research was conducted as part of the honors requirements for Middle Tennessee State University. My name is Shelby Cox, and I'm a pre-medical student that just graduated from MTSU actually. So everyone knows that cannabinoid products are the craze right now and that they have many health benefits. Many have been found for a variety of illnesses, including multiple sclerosis, acute paranoid schizophrenia, psychosis. And this new generation of cannabis farmers prefer plant propagation instead of growing plants from seeds because it's been proven to be the most cost-effective method to growing, in theory, genetically identical plants that will have consistent rates of growth and cannabinoid production. Especially when compared to plants that were grown from seeds. So plant propagation is when you take a tissue culture from a stock plant and you use that plant cutting or that plant tissue to grow a bigger plant. However, recent studies have shown that plant propagation can lead to certain genetic changes known as somoclonal variation. This could prove problematic for farmers who use plant propagation because sometimes they try and preserve or retain certain cannabis attributes through many generations. This project measured and compared the cannabinoid levels of 3 different varieties of hemp through multiple clonal generations. The varieties were named Cherry, Cherry Blossom, and Cherry Workhorse. So we took cuttings of cherry plants and we let them grow into larger plants, and then we took cuttings from those plants and grew them into larger plants, and then we took cuttings from those plants and grew them into larger plants. We did that 2 or 3 times, and we did that for Cherry, Cherry Blossom, and Cherry Workhorse. I hope that makes sense. And as you can see in the following 3 graphs on the poster, many cannabinoid levels were significantly different between generations. This is interesting in itself, as obviously offspring, in theory, should have been genetically identical to the parent plants. But it's also interesting to note that there are many large differences between the average cannabinoid levels in generations, but the differences were not significant. So there were differences, but they were not statistically significant. This was caused by a few plants in each generation that had such high levels of a certain cannabinoid that it pulled the average extremely high, whereas other plants had absolutely none of that cannabinoid in the analysis. So for an example, it can be seen in the first graph named cannabinoid levels of Cherry Workhorse. For the cannabinoid THC9, if you look at the second generation that is in orange, it had absolutely no THC9 at all, but some plants in generations 1 and 3 had lots of THC9. It's important to remember that these points on the graph are not significant, as there's not a star at the bottom indicating significance, but a few plants had high enough THC9 that even when averaged with many zeros, the average for that generation was still very high. So just a few plants had enough THC9 to pull the average up way, way high. So it wasn't statistically significant because there was not a large difference over the entire generation. It was just a few plants that pulled the average that high. But as we had said previously, they should be all the same, as they're supposed to be genetically the same because they came from the same plants, which is obviously not true. This study shows that there is a potential for somoclonal variation in cloning of hemp that affects cannabinoid levels. I hope if any of these topics interest you, then you are more than welcome to join us in the meeting room. My name is Shelby, and I hope to see you there. Also, if it is difficult for you to read any parts of the poster, I have added a downloadable PowerPoint version of the poster under Uploaded Resources. Thank you.